The immune system, innate and adaptive body defenses. The nonspecific body defenses have surface membrane barriers. The first line of defense is the skin. It is a barrier to pathogens, and the oil that's on the skin inhibits the growth of pathogens. You also have the mucous membranes, which those are good places for bacteria to get into your body. You have the eyes, and those use tears to wash away bacteria, and blinking helps keep bacteria out. And the nose and the respiratory tracts have hair to trap the dirt, and the cilia moves all the mucus from the trachea and the bronchi. And then you have the GI tract, which you have the acid in the stomach that kills most bacteria that gets in there. And the population of bacteria in the intestines are crowded out. The nonspecific cellular and chemical defenses, and then you have the second line of defense, the phagocytes. And the macrophages are at the site of a chronic infection. You have free macrophages which wander throughout the body and dust cells in the lungs and dendrite cells. Fixed macrophages ingest foreign materials and recruit more macrophages. And another phagocyte is a neutrophil, and that's at the site of acute infections and are the first responders. They have defensins, which are produced by neutrophils and poke holes in the target cells. The second line of defense continued. You also have the natural killers, which release perforins that poke holes in cell membranes and promote inflammation. And signs of inflammation are redness, swelling, heat, and pain. As you can see on the hand right here, you can see the swelling, you can see the redness, but you can't see the pain, but thanks. The complement system. This helps kill bacteria, and it consists of 20 different proteins. Some actions of it are direct lysis of pathogens, which that means it tears open pathogens. Opsonization, is adding handles to pathogens to make them easier to grab. So it's just like a suitcase, so where you can just pick it up. And the complement system also aids in the inflammatory response. And all these three things happen at the same time. Specific biodefenses. Third line of defense, you have characteristics of the immune system. Antigen specificity only works against particular antigens. Then you have systemic, which is active throughout the whole body. And memory, which is after the first response. The next responses are stronger and faster because they already know the pathogen. Some cells of the immune system, you have lymphocytes, which come from red bone marrow and need to bind to an antigen to be able to do its job. And they become immunocompetent. The T cells become immunocompetent in the thymus, and the B cells become immunocompetent in the bone marrow. The humoral immune response, which is the primary immune response, you have the clonal selection, which antigen attaches to B cell. The B cells get help from helper T cells. B cells begin to divide and differentiate and make plasma and memory cells. And with the plasma cells, they are factories for antibodies, and they generally last 4 to 5 days. And the memory cells, which I said earlier, remember the first attack and make faster antibodies that last longer. Now you got the active immunity and passive immunity. In active immunity, your body makes antibodies, and when they're naturally acquired, the natural process, you get sick and you get better. Artificially acquired is you get a shot or a vaccine. Passive immunity is antibodies come from somewhere else. So in the naturally acquired section, antibodies come from your mom when you're born. And artificially acquired, you get through a needle as well. And this is the antibody. This is the structure of the antibody. You have 2 heavy chains and 2 light chains, which the 2 heavy chains are that pink area, and the light chains are the green. You have the constant region, which is just that little part, and then the variable region, which is where the antigen binding site is. And stop.
This is an IgM antibody. It's called a pentamer because it looks like a pentagon. There's 5 antibodies that go around it. It's the first antibody produced by plasma cells in a primary immune response, and it readily fixes and activates the complement system. This is an IgG. It's a monomer because there's only one antigen. It's found on the B cell surface and it functions as a B cell antigen receptor.
Okay, this is a dimer. It's called a dimer because it has two antibodies attached to it, and it's a secretory antibody found in body secretions, such as saliva, sweat, intestinal juices, and milk, and it helps stop pathogens from attaching to epithelial cell surfaces.
This is an IgE. It's a monomer because there's one antibody. It's the rarest of all, and the stimin binds to mast cells or basophils. It's an antigen binding to its receptors and triggers these cells to release histamine and other chemicals that mediate inflammation and allergic reaction. And only traces of IgE are found in plasma.
Alright, this is a IgG. It's a monomer just because it's one antibody, and it's the most common antibody. It's accounting for 75 to 85% of antibodies, and it's produced by plasma cells in an immune response. And it readily fixes and activates the complement system, and it protects against bacteria, viruses, and toxins circulating in blood and lymph. The cell-mediated immune response, you have cytotoxic T cells, which attack cells affected by bacteria, helper T cells, which help activate T cells and direct the adaptive immune response. Then you have suppressor T cells, which turn down the immune response. Alright, so here we start out with the pathogen, and the pathogen is floating around your body, and a macrophage comes along and eats it, as you can see here. And then macrophage then chops up the pathogen, and then it becomes an antigen presenting cell, so where the antibodies are showing off the pathogens on the end of the antibodies.
That antigen presenting cells attach to the helper T cells, which then they divide and differentiate into active helper Ts and memory helper T cells. And memory helper T cells just kind of go away and come back when they're needed.
Alright, so active T cells, helper T cells, use cytokines to trigger B cells, which then attract all the pathogens. As you can see here, the B cells have antibodies on the end, which are each of these. And once the B cells get pathogens on them, they divide and differentiate into memory cells and plasma cells, and those memory cells go away for later, and the plasma cells are responsible for making all the antibodies against the pathogen.
The antigen presenting cells and the helper T cells all connect to the cytotoxic T cells, which then comes down here, and they divide and differentiate into activated cytotoxic T cells and memory cytotoxic T cells. And the memory cytotoxic T cells just kind of go away and come back when they're needed.
Alright, so all of these are normal body cells, and the normal body cells with the orange in them, those are pathogens, so those are being affected by certain pathogens. And you have the proteins down here that are showing if they're affected by pathogens or not. And the cytotoxic T cells know where to go by looking at these proteins, and they attach to the body cell and kill the pathogens inside. Now we're going to get into some organ transplants. So next. You have the autograft, which is a transplant from yourself. So as you can see in this picture, this is a skin graft. So they're taking skin from somewhere else on your body and putting it where your wound. This mostly occurs with burn victims. Then you have an isograft, which is a transplant between identical twins, and that's not very common, but it happens because with identical twins they have the same DNA and genetic structure, so that's why that happens. Next. And you have an allograft, which is a transplant between same species, so that's when you get on a transplant list and you're waiting for someone to donate an organ to you. So that can go from human to human or dog to dog, but it has to be the same species for allograft. And you have the xenograft, which is a transplant between different species. So you can see over here in the human, you have red blood cells that are being put into the mouse, and that's just because it's different species. Some homeostatic imbalances of immunity. You have the hypersensitivities, they're allergic reactions, and they're organized by how fast they happen. You have the immediate type hypersensitivities, which happen within seconds, and the antigen binds with IgE, and basophils and mast cells release histamine, which cause inflammation. And you have subacute hypersensitivities, which begin within a few hours, and cytotoxic reactions, which occur with mismatched transfusions with those organ transplants if they don't match correctly. Then you have the immune complex reaction, which means you have too much antibody and antigen complex. Some examples of that would be lupus or rheumatoid arthritis. Then the delayed type hypersensitivities, which take a couple of days to occur, and that's like contact dermatitis, which can become from poison ivy, and also graft rejections, because they could take a couple of days to happen.